Hello, everyone. Welcome. Just going to wait another couple of minutes to let some people join in, and then we'll get started at 11. All right. And welcome, everyone. Hello. Thank you all for joining today. Um, hello, everyone, and welcome to the Invasive Species Center webinar series. My name is Vince Latassa, and I'm a program development intern here at the Invasive Species Center. I will be your moderator for today's webinar. So before we begin, I would like to acknowledge that the Invasive Species Center honors the long history of the First Nations, Inuit, and Métis Nations people on the lands now known as Canada, and strives to show respect to their ancestors, their communities, and to them individually. We greatly appreciate the significance of the lands, waters, and all living things, and offer our gratitude to the Indigenous people for their care and teachings about our Earth. Our relationship with the Indigenous communities are important, and we will continue to listen and learn how we can be in good relationship with Indigenous peoples, the lands and waters, and all living things, and act accordingly. The Invasive Species Center respectfully acknowledges that our head office is located on the traditional lands of the Anishinaabe, the Batchewana, and Garden River First Nations, as well as the longtime settlement of the Métis people in the Robinson-Huron Treaty area. So the Invasive Species Center is a non-for-profit organization that mobilizes action against invasive species that harm the environment, economy, and society. Our website is full of valuable invasive species resources, including best management practices, species profiles, video resources, and much more. They're all free to access, so be sure to check us out at www.invasivespeciescenter.ca. You can also sign up on our homepage for our official newsletter titled The Spread, our bi-weekly media scan, and event invitations, which is where you will hear about our upcoming webinars and keep you up to date with the latest on invasive species. The Invasive Species Center has launched a new invasive training program that offers virtual courses on topics related to invasive species. We currently have 3 courses available, but we'll be releasing new content regularly, so stay tuned for that. Make sure to check out our website and sign up to receive updates on when these new courses will become available. Anyone with questions can check out our training program website FAQ page, or you can also send us an email at training@invasivespeciescenter.ca. Before we get started with today's webinar, there are a couple things that I'd like to go over. There will be time for questions at the end of the webinar, so if you have any questions at any time, please type it in the Q&A box, and I will read it to our presenter at after the webinar. If you are having technical difficulties at any time, please type them in the chat box or respond to the email found in your registration, and we will do our best to resolve them. Closed captioning is available for anyone wishing to follow along that way, which can be enabled by clicking the closed caption button at the bottom of your screen. Lastly, there will be a very brief survey following the webinar. If you could take some time to fill it out at the end of the webinar, it would be greatly appreciated. So today's webinar is titled Solving the Mysteries of Beech Leaf Disease, 6 Years of Partnerships, and will be presented by Sharon Reed. Dr. Sharon Reed is a forest health research scientist and serves as program lead for the Ontario Ministry of Natural Resources and Forestry. Sharon's research program focuses on the detection, impact, and management of invasive forest insects and diseases. Sharon also provides scientific support for the Provincial Forest Health Monitoring Program. Dr. Reed's research topics include oak wilt, beech leaf disease, hemlock woolly adelgid, spongy moth, and phytophthora. Over the past 6 years, Dr. Reed has worked collaboratively with international partners to identify the cause of beech leaf disease, its impact on Ontario forests, and to improve detection methods. Currently, Dr. Reed's research is focused on identifying how the beech leaf nematode is transmitted locally and across long distances. We are very fortunate and excited to have Sharon with us today. So without further ado, I am pleased to introduce you to our speaker, Sharon Reed. Thank you, Sharon, and I'll pass it over to you.
All right, thank you, Vince. All right, I just want to make sure that you can see the full PowerPoint presentation.
Yep, it looks good.
Okay, great. All right, before I get started with this presentation, I want to acknowledge the staff in the Forest Health Research Program. I will be presenting some of the data that they've produced this past year. First of all, Dr. Namatar Shingandu and Dr. Endurance Awere. They both are doing molecular work in the lab for both pathology and entomology. Brittany Cote has led the field work in our lab and is currently summarizing the data from the beech leaf disease, beech bark disease plots, and Dr. Violet Butterworth, who helped with the data collection this summer. So today's presentation, since this is a mixed general audience, I'll be covering the symptoms of beech leaf disease, talking about the causal agent, a nematode. I've included some information that was published recently in 2023 about beech leaf disease that's important to understanding this disease, and as well as the new range map that was published in December, and some information about the distribution of the nematode in Ontario. I'll finish up talking about the beech leaf disease, beech bark disease plot network, which is an international collaboration that is now in its 5th year, going on 6th, and also an international joint venture to understand how the beech leaf disease nematode is being transmitted. So I'm starting my presentation with a timeline, just to give you a sense of how rapidly our knowledge about beech leaf disease is progressing. The first detection was made in Lake County, Ohio. That's the 2012 point on the far left of the timeline. But in 2017, that was when we first determined that the nematode and the beech leaf disease was present in Ontario, with detections of the disease in Elgin and Norfolk County. The year after that, in 2018, the first meeting about beech leaf disease was hosted by the USDA Forest Service in Ohio. And during that meeting, research and monitoring task groups were formed. And those groups really accelerated the knowledge about beech leaf disease and the research to understand it. The following year in 2019, the beech leaf disease, beech bark disease plot network was established. And by 2020, there was a joint paper that described how the nematode was consistently associated with the beech leaf disease symptoms, and described the nematode and the symptoms that could come from nematode feeding. In 2021 and 2022, molecular markers had been developed and published for the nematode, making it easier to identify if this particular nematode was present in a sample. And in 2023, several important papers were published on the effects of nematode feeding and the effects of beech leaf disease on leaves. And by 2023, it was already the 5th year for the plots in Ontario to look at the impacts of beech leaf disease and beech bark disease together. Right, so beech leaf disease affects American, European, Chinese, and Oriental beech. It only occurs in Eastern North America, and I'll be showing you slides of the range in a few minutes. It causes the decline and mortality of understory and overstory trees, and the only known causal agent is a foliar nematode Phytolenchus crenate mccannei. It is the infestation of the buds by the nematode that results in the leaf damage, and that's a really important concept to remember when you're dealing with beech leaf disease, that the damage happens in the buds and there's no progression of the symptoms over the growing season. Right, so the symptoms of beech leaf disease, the main characteristic sample, the photo on the top left shows those dark green bands. Sometimes they're described as like slightly swollen dark green bands in between the leaf veins of the beech leaf. And those bands, they can look a little bit different depending on the time of the year. So the next picture over to the right, that is a leaf that just leafed out. So the bands have a slightly reddish color sometimes at the very beginning of the growing season. And then on the far right, on the top row, there's some fall foliage leaves where those dark bands stay dark or turn black, and the rest of the leaf is taking on that yellow fall foliage color. When the symptoms are more severe, you get a picture like the one that's more in the middle of the top row or the bottom left, where the leaf becomes more thickened and leathery. The leaf can curl, and sometimes these are called crinkled leaves. And they can get this yellow chlorotic look to them as well. Other symptoms of beech leaf disease, one would be early leaf drop. Some of these severely affected leaves can start dropping as early as June. Then there's also bud abortion, where buds, because of feeding damage or because the branch is dying, are aborted. So we don't talk about nematodes very much in forestry because there are very few diseases that are caused by nematodes for trees in natural forests. But these nematodes, they can cause physical damage when they're feeding. Sometimes they inject enzymes and metabolites that alter cell function or kill cells. There's some nematodes that transmit bacteria or viruses while they're feeding, and their feeding can create wounds that allows other pathogens to invade those damaged plant tissues. And a lot of times with nematode diseases, the size of the nematode population is important to consider. So this past year, Dr. Vera and his colleagues published a paper that talks about the damage caused by the nematodes feeding on the bud scales and the leaf primordia. They found that nematode feeding increased reproduction of cells, the size of the cells, the cell layers, and the number of chloroplasts. And this resulted in an extremely disorganized structure in the leaf tissues. So on the photo on the bottom right, photo F, is one where you can see there's just a whole bunch of cell layers that you wouldn't normally see that many cell layers in the cell. And you know, they're just very disorganized, not neat rows that you would normally expect to see. Also in 2023, Dr. McIntyre produced a paper talking about the effects of beech leaf disease on leaf physiology. So this was a small study just at one location with 12 trees, but he compared the leaves with a little bit of banding to the extremely affected crinkled leaves to asymptomatic leaves, and found that the symptomatic leaves had reduced carbon assimilation, less instantaneous water use efficiency, less specific leaf area, and had more stomatoconductance, leaf water content, nitrogen content, light compensation, and dark respiration. So that's a mouthful, but what does that mean? So a thicker mesophyll coupled with a smaller total area results in a denser and more hydrated leaf, which could benefit the nematode. Increases in dark respiration suggest that stomates may not be properly closing. And symptomatic leaves exhibit significantly lower rates of carbon assimilation compared to neighboring asymptomatic leaves. So the implication of this points towards a compromised tree carbon budget, which can negatively affect growth and vigor of the tree long term. Another interesting publication that came out was a master's thesis from the lab of Enrico Bonello by Salvi that identified risk factors associated with beech leaf disease development. So they found that the difference between the mean temperature, the warmest and coldest month, was an important predictor for risk, with temperatures between 24 and 20, temperature differences between 24 and 26 degrees Celsius having the highest risk. And that coupled also with high relative humidity between 56 and 76%. And you'll probably see in this presentation, there's a little bit of theme that water is an important component for foliar nematodes. They also found that disease incidence was greatest between 0 to 200 kilometers of major water bodies and near riverbeds. Other important factors included soil bulk density, high soil nitrogen content, higher elevations, high autumn and low spring precipitation, and terrain ruggedness index. So as I was saying earlier, a new map for the distribution of beech leaf disease was produced this December. So the map I'm sharing with you can be found on the Cleveland Metro Parks website. I've included it here at the bottom of the slide, but if you just Google Cleveland Metro Parks and publications, you will find a link to this map. So there has been expansion. All of the counties in orange are new detections for 2023. You can see that there were small increases to the south, west, and to the north of all of the known locations where beech leaf disease has been detected. So from Michigan to Virginia up to Maine. And in Ontario, we had 4 new counties. 3 of those counties were just slight extensions of the range already, but the detection in Simcoe County was more of a jump across the landscape than we were expecting. So my lab has also been tracking the distribution of the nematode. We've been doing this by having our biodiversity monitoring section, the forest health research monitoring section, send in samples to our lab, as well as some researchers and other individuals that are interested in determining if the nematode is present. So over the 2 years, we've had 354 samples sent into us, and all of the samples that had beech leaf disease symptoms were positive for the nematode. And you can see those locations from the past 2 years on this map, all the purple dots. What is interesting is that we also have a number of asymptomatic samples coming into the lab. And the green ones are asymptomatic samples that were negative for the presence of the nematode DNA. But the orange are ones where we detected nematode DNA in the sample. So the nematode has been detected in samples as far west as St. Joe's Island and as far north as Algonquin Park. We'll continue to watch these areas to see if beech leaf disease does develop in this area. Just because nematode DNA was detected there does not mean that the disease will develop. It could be that some of these nematode populations are very small and may not be favored by the environment. So now I'm going to talk about the first of the two international collaborations that I will talk about today. So we have been monitoring the spread and impact of beech leaf disease in Ontario forests. In 2019, plots were set up at 17 locations. So each one of these locations on this map along Lake Erie, there's 2 overstory plots, there's 2 plots for saplings and 8 plots for seedlings. And the type of information that we collect in these plots include overall dieback, crown density, main and fine branch dieback. We rate the severity of the beech leaf disease symptoms, as well as the severity of beech scale and neonatria canker. And for those of you not as familiar with diseases of beech, the reason why we are rating the scale and the neonatria canker is because when beech scale feeds on beech trees, it can allow neonatria to invade the bark tissues and cause beech bark disease, which can kill overstory trees. For the seedling plots, we record the different woody species that are in those plots and their percent cover, and we record the number of seedlings that have beech leaf disease. When this study was set up, we wanted to, compare different conditions. So we had 4 locations where the trees only had beech bark disease. We had 5 locations with beech leaf disease and beech bark disease. And we had 8 locations with beech leaf disease. And some of those locations had severe beech leaf disease already, and others had very little beech leaf disease at that time. So I'll be showing you how that changes. But first, I want to point out that the plots in Ontario are just part of a much larger plot network that extends throughout the eastern United States. The plots that were set up in 2019 were set up in Ontario, New York, Pennsylvania, and Ohio. And the rest of the plots were established later on. Okay, so this is the first slide for the 2020 results. So again, this is the 5th year of this study. The first thing that's I've noticed is we'd only have 2 colors now instead of 3 colors. So beech leaf disease has now developed at all of our locations, study locations. So we no longer have plots that just have beech bark disease. We also had beech bark disease develop at 4 sites that didn't have the disease earlier. So the study is really becoming a comparison between locations that have beech bark disease and beech leaf disease versus locations that have beech leaf disease. So for the overstory trees, we've been rating 240 beech trees. These are individuals that have a diameter at breast height over 10 centimeters. And we've seen an increase in beech leaf disease. In 2019, we had around 50% of the trees infected with beech leaf disease. Now it's 85% of those trees infected with beech leaf disease. From the 7% that had beech bark disease in 2019, now it's 19% of those trees. And the percentage of trees that have both the diseases has gone from 3% to 17%. And we had 14% mortality of the trees that we've been rating over the 5 years. For saplings, we have 497 saplings that have been rated. These are individuals that are tall enough to, they're at least 1.3 meters tall, but they're less than 10 centimeters in diameter at breast height. So again, there's been an increase in beech leaf disease. We've gone from 41% of the saplings infected to 89%. There's no trees, no saplings with beech bark disease, and that is typical for beech bark disease. It typically only affects overstory trees. And we had one tree in 2019 that had beech leaf disease and beech bark disease, and that tree has died. So now we don't have any trees in that category as well. For the saplings, we've recorded 22% mortality. We also look at dieback. So this is a visual representation of what we are observing across the landscape. So the red, those are locations where there's very high dieback, greater than 67%. Moderate dieback, the orange, that's 33 to 67%, and yellow is less than 33% dieback. So visually, you'll notice that most of the locations that have moderate to severe dieback are located around the area where we first detected beech leaf disease, so around Norfolk and Elgin counties. There's an increase in dieback at all locations, but it's just more severe in these areas where beech leaf disease has been present the longest. And we've also seen beech bark disease uptake there. For the saplings, really the same pattern, except that there's more spread across the landscape away from that original detection with the moderate severity sites at Sinclair, Longwoods, and Lawson. Right, so when we started the study in 2019, we had trees in size classes, trees and saplings, sorry, in size classes from less than 1 centimeter DBH to greater than 60 centimeters DBH. When you look at the trees that have died, trees and saplings, the majority of that mortality has been occurring in saplings that are less than 5 centimeters DBH. There is mortality occurring in the other size classes, but it's just so small that it's really hard to see it on this graph. And when we started this study, we already had heard reports that beech leaf disease affected saplings, causing mortality. So we wanted to better understand the effects of beech leaf disease on forests with and without beech bark disease. And again, beech bark disease occurs when the feeding by the scale creates wounds that allows a fungus to invade the tissues, and a lot of these little cankers that develop right next to each other can shut off the sap flow in the tree and cause mortality. And when those trees die, they send up a whole bunch of sprouts from their roots, which can cause what we call beech thickets that crowd out other types of trees that might be more healthier and vigorous in the long term. So at year 5, this is our first glimpse of what the differences between these different forests that are affected by 2 diseases versus 1 disease. So when beech leaf disease is only present, we see much lower mortality of overstory trees, just 7% so far. And the sapling mortality is much higher at 17%. When both diseases are present, then there's a much greater increase in the amount of overstory tree mortality. We have 20% mortality right now, and sapling mortality is 26%. So with both diseases are present, it seems like we're just seeing the slow removal of beech from these forests. Okay, and so then the last project I wanted to talk about was the transmission of the beech leaf nematode. And as I said earlier, this is in a national collaboration. So there's a question, we see beech leaf disease developing so quickly across the landscape, especially compared to other diseases. So we want to know, how are these foliar nematodes moving around? So foliar nematodes exit and enter leaves through the stomatas and wounds, and they move in water films. This is really important for foliar nematodes. So rain and humid conditions create these water films that allow nematodes to move across plant parts. And there are some nematodes that also could be blown in the wind. And we want to know, could these nematodes be crawling on the plant surface and become entangled with insects or mites or just any arthropod, then be transferred to another tree or location? And or could they even be digested and survive the digestion? Also, can nematodes be found in flower buds, the parts from the flower buds? Can they be found in nuts? Could they be found in, we know that they occur in old leaves. Is there a way that they'd be able to leave those old leaves after being blown around in the wind and infect another tree? So we've been awarded an international joint venture funding from the USDA Forest Service that's going to allow us to work with Dr. Mihail Cantor from the Pennsylvania State University, as well as Danielle Martin and Cameron McIntyre, to investigate some of these questions. So one thing that our lab had already been doing some work on was the collection of mite and arthropods. And we collected mite and arthropods from 3 locations that had symptoms and 3 locations that did not have symptoms. And so for every plot, we were collecting 20 leaves 1 time per month from June to October, and looking at these leaves under the microscope, collecting the arthropods that we found, identifying the arthropods using molecular markers, as well as nematode DNA, and then also using the DNA to confirm if the nematode was present in the leaves. These are examples of some of the pictures of the mites that we found. They all look the same to me, but when we blast them, they come up as different individuals. So these on the far left is a tided mite, a Tentranchidae, a spider mite, is next to that. Then a predatory mite called Agastemus lobatus, and also on the far right, a predatory mite called Phytosciidae. So far, we've been able to identify the arthropods from 2 of the 6 sites. 1 site had beech leaf disease, the other one did not. There were 32 taxa that were identified. And the most common arthropods were the tided mite, which is kind of a bit of a generalist, could be predatory. Some are fungivores, some are scavengers. There's a Phytosciidae species and a Tentranchidae species of mites. There's also was an aphid that was common, this Phalaphus grandifoliae, and the tided mite was more abundant at our site that had beech leaf disease, and the aphid was less abundant. So we're currently working on the other 4 sites. And it'll be interesting to see if we see any differences in the community between the symptomatic sites and the asymptomatic sites. The selection process that we used really favored small arthropods previously. So this August, we made a collection of large arthropods at Chapman and Kirk Cousins. And we collected these large, mostly insects from the crown of sapling trees, as well as from tree boles. We ended up with 134 individuals. We also collected another 11 caterpillars from the nearby vegetation. These are just photos of some of the caterpillars that were collected. Most of them were tussock moths. Under the direction of Dr. Mihail Cantor, we investigated the possibility that there could be nematodes in the caterpillar frass. So we reared the caterpillars in plastic food storage bags and collected their frass. They had been feeding on the symptomatic beech leaf disease samples. And we placed that frass in these cups with a netted bottom, and then we soaked the frass to see if nematodes swam out of the frass. All right, so we found dead nematodes in 7 out of the 12 samples. They were intact, but we didn't find any live nematodes this year, so we were a little less successful than our counterparts. But we are planning on repeating this next year, hopefully to collect more insect or more caterpillars, hopefully larger caterpillars than we had this year. The other arthropods that we had collected, we washed the outside of the arthropods and tested the rinsate for the DNA of the nematode. Then we ground up the arthropods and tested the macerates. We ended up with 4 positives. All of those were from the ground up macerate. So we will be further investigating this next year to see if these larger arthropods could be vectors of the nematode as well. In this year, since we didn't really get started on this project in August, but we were still able to collect some seeds. The biodiversity monitoring section sent us some viable seeds, and Endurance soaked the nuts as well as the shell and visually screened them for nematodes and was able to find a live nematode in one of the seeds. So we will be continuing this next year to see how often this occurs. Okay, so just to summarize up the key messages from the presentation, there have been a large number of partnerships and international collaborations over the past 7 years that have really quickly advanced our knowledge of the disease distribution and impacts. The beech leaf disease is the beech leaf nematode, Leilancus crenati mecanae, is the only known causal agent of beech leaf disease. And recent research has explained some of the mechanisms for symptom development, as well as the effects of the changes in the leaf. We are seeing an increase in beech leaf disease in Southern Ontario, as well as beech bark disease. Beech leaf disease is still more widespread than beech bark disease in the study area along Lake Erie. But both of these diseases together are having a severe impact on the forest, both the overstory and the understory. And last but not least, the investigation of how the nematode is being spread around from tree to tree and location to location is still ongoing, and arthropods and plant parts could play a role. And with that, I'll take questions.
All right, awesome. Thank you so much, Sharon. So we do have a couple of questions here in the Q&A that I'll read out to you. So the first question that came in here from Dana says, of the 354 beech leaf samples submitted for eDNA testing, who submitted those samples? And were those collected at random by your colleagues or submitted by partners?
So the majority of the samples were sent in by the Forest Health Research Monitors. They're part of the Biodiversity Monitoring section of MNRF. Some of the researchers here that have beech in their study areas also sent in samples. So mostly individuals associated with the Ministry of Natural Resources and Forestry. But we did have a few samples sent in from individuals from conservation authorities.
Okay. And we have another question here. How many generations of nematode from bud break to bud formation?
From bud break to bud break. I don't know the answer to that question. I do know that some nematodes can complete a generation within a month's time.
Okay. And just a quick reminder to those who are asking questions to put them in the Q&A chat box, not the chat box itself, but the Q&A one. There's another question here that's asking, how do you manage the dense beech thickets that form after tree death?
So there is ongoing research by another scientist at the Ministry of Natural Resources and Forestry that is looking at the management of these beech thickets, looking to see if chemicals can be used to kill some of the beech seedlings or if some type of tending process can be used.
Okay. Another question here. Will there be follow-up research on the impacts of live nematodes present in the beech nut on seed viability?
I think for my lab, the next step will be, as I said, looking to see how common it is for the nematode to be found in the beech seeds, as well as if it's possible for the nematodes that are in these beech seeds to somehow reinfect a healthy beech seedling or beech sapling. So I don't have plans for doing that research, but there may be others out there that are looking at that question.
Another individual here is curious to know if any inoculation studies have been conducted.
Inoculation studies for the beech leaf nematode. So back in, I think it was probably 2019, 2018 possibly, their inoculation studies with the nematodes were done, and the inoculations of the beech buds did result in beech leaf disease symptoms.
Another question here. Are beech and colder mountain climates generally safe?
That is a great question. I do not know the answer to that question. That might
Okay.
be a question for Enrico Bonello's lab.
Okay. Another one here. Someone wants to know if the treatments for beech leaf disease are similar for the treatments for beech bark disease, including chemical spray and removing thickets. So just are the treatments similar?
So there are no approved treatments for beech leaf disease. There are some treatments that are in an experimental phase, especially two that are being tried in the US. One acts as a fertilizer, and the other one is a nematicide that kills nematodes.
Okay. And is it recommended, sorry, the question went down. Is it recommended to avoid planting beech trees at this time if a jurisdiction has found the nematode present?
I there are no recommendations not to plant beech trees. Um, there, something to remember that all of the plots that we have that we are studying beech leaf disease with are in forest settings where the canopy is more dense and there's more humidity. So, one of the questions would be, how are open grown trees more likely to survive beech leaf disease or avoid infection by the nematode. So I don't think the research isn't really there yet to make a recommendation for or against.
Okay. Um, Another question, any estimation of the total impact of beech leaf disease and beech bark disease on the beech trees within the Great Lakes? So are we looking at something like ash borer or Dutch elm or slower?
Right. So was the question, is beech leaf disease faster or slower than Dutch elm disease?
I'm assuming that's what's being asked, yes.
Yeah. So the development of the disease across the landscape, I would say, has definitely been faster than other diseases that um, we've seen. It's more similar to some of these airborne diseases like chestnut blight. But I think one of the unanswered questions is, the nematode, how widely is it distributed already? Um, and how long has it been in these areas before we could really say, which disease is spreading faster?
And there's another question here in relation to the transmission pathway. So it says, what is the suspected transmission pathway for long distance movement of the nematode beech leaf disease? Is it the movement of soil, birds, nursery stock?
So that is that's the one of the questions we're trying to answer right now. So we don't know what are the major routes of transmission right now.
Okay. Um, Someone else here had asked, why are younger beech trees more affected than larger overstory trees?
So again, not something that I can quote any research on. Um, it likely, since it's a foliar nematode, has to do with the condition, um, the more dense, humid environment in the understory may be playing a role in the fact that there's more beech leaf disease. Another factor could be that these young beech have less carbon resources to withstand the disease. So that might be why mortality is higher there as well. Um, we've definitely observed over the past 5 years that with the overstory trees, usually the bottom of the half of the tree is more severely affected by beech leaf disease, whereas the leaves that are up on the top of the tree that are more exposed to the sunlight, they have less beech leaf disease typically. So there could be some more environmental effects there.
Okay. Um, Is there any recommendation for harvesting and selection of beech for saw logs and firewood?
So I can put my email in the chat for that individual and send them a information report from the ministry. Um, so so if they're willing to share their email, I can share that information. Um, we do not promote the cutting of healthy beech trees, only beech trees that have beech bark disease or have a lot of deformities on them already, because the deformed trees are more likely to be infested by a scale insect and develop beech leaf or beech bark disease later on. There are no recommendations associated with beech leaf disease.
Um, Another question here. Are there any strategies for informing the public regarding these threats to the beech trees? And how can local organizations monitor their trees in an effort to contribute to the science?
So we currently do not have any citizen science projects right now. My lab has been accepting potentially symptomatic samples in our lab, and then we have been testing them and then letting people know if beech leaf disease is present or not present. So that's one thing that could be done if you suspect that beech leaf disease is already present in your area or on your property. But otherwise, in Ontario, there are no citizen science projects. It could be an
Okay,
area for an
and
invasive species center.
right. um, this other question here. So, will the beech leaf and beech bark disease render the American beech functionally extinct, similar to the American chestnut?
We really cannot answer that question with just 5 years of data at this point. There are trees in Ohio that have been reported to be resistant to beech leaf disease, and we know that there are trees that are resistant to beech bark disease. So I would suspect, I'm going out on a limb here, to say that there will be beech on the landscape, but far fewer than we've had in the past.
Okay. And there have been a couple of questions here in the chat asking um, about the origin of the nematode, whether or not it's native to North America or if it was introduced.
So Lidolanchus crenate has been reported from Japan, and all of the Lidolanchus species that are known are from the Pacific Rim region. So right now the assumption is that this is an invasive species from outside of North America.
Okay. There's another question here. One of the slides had soil nitrogen contributing to the nematodes. So I guess they're asking whether or not fertilizing is a bad idea.
So, um, so yes, that was one of the soil nitrogen was a risk factor for beech leaf disease. Um, I think what I would say here is that you need a suite of factors for beech leaf disease to develop. And it's not necessarily going to be fertilization that's going to create a situation where beech leaf disease could develop.
Okay. And another question here is there a threat of moving nematodes picked up on boots when hiking in an infected forest to a non-infected forest?
So we are recommending that individuals clean their boots after being in a forest that has beech leaf disease. And, I would really recommend that for anybody, regardless if beech leaf disease is present or not. There's just a lot of invasive soil pathogens that can be, carried from one place to another without us realizing it. So I would definitely encourage boot cleaning.
Right. And that's something that we also encourage as well from the Invasive Species Center, just for invasives in general. If you are in an area that there are known infestations of invasive species, especially plants and smaller insects, that definitely like clean your boots and gear. So it's a good question from the person who submitted that. Um, Perhaps maybe another one here. Are these nematodes associated with tree disease in their region of origin? For example, the Pacific Rim countries.
So in Japan, the Lidolanchus crenate nematode can cause some banding symptoms on leaves, but it's not, like, it could be a little bit of galling, but it doesn't develop into a disease that affects the Japanese beech trees.
Okay. Another person here had said, so saplings in humidity, lower limbs on overstory. How about removing lower limbs on the overstory trees?
Um, that's an area that needs to be researched. If any alterations to the stand could reduce the amount of beech leaf disease, be it thinning or reduction in limbs.
Okay. So I think that covers most of the questions that were inputted here. So before I do my little conclusion, or were there any other concluding thoughts or last remarks for the audience?
No, just thank you for everybody who attended today. I'm really happy that I was able to share this information with you. And I'll check back with you in 5 years about these beech leaf disease, beech bark disease plots, and hopefully we'll have some very clear answers for you at that point.
For sure. And maybe if I'm not sure if you can provide that in the chat, but if you'd like to input your email, if any of the attendees have questions and would like to reach out to you after the webinar is over, that would be great as well.
Let me see if I can get back to that page. I think I need to stop sharing in order to do that. Okay. Yeah, so I'll put it in the Q&A chats, my work email. Well, actually, where is the chat? I'll just tell you, it's sharon.reed@ontario.ca. And I don't know if you would be able to put that in for me.
Yeah, so I inputted that
Thank
into the
you.
chat box
All
there
right.
for you. So if anyone is looking for the email, just check the chat box and you should be able to see it there. Okay, and I'm just gonna pop up my display. All right. So thank you again, Sharon, for the presentation. It was really interesting. I learned a lot from it, and I'm hoping that everyone in attendance did as well. So just so everyone knows, this webinar was recorded and will be posted both on our website at www.invasivespeciescenter.ca and also on our YouTube channel. And just a friendly reminder to please take a couple of minutes to fill out the survey after the webinar has ended. We would really appreciate that. So next month, the Invasive Species Center will be hosting our annual Invasive Species Forum from February 12th to the 15th. So there will be no regularly scheduled webinar. The Invasive Species Forum is an annual event that brings attention to invasive species issues, research, and advances in prevention and management occurring across Canada and the neighboring United States. So anyone who is interested in attending the forum, you can register for free through our website at invasivespeciescenter.ca/forum. And we have a really exciting lineup of speakers and engaging topics this year, so be sure to register to secure your spot. And lastly, our webinar series will resume in March with a presentation given by Tricia Tugay, who's an aquatic biologist with Fisheries and Oceans Canada. She will be giving a talk on the grass carp captures from the 2023 surveillance season. And this webinar will be on March 7th at 11 a.m. So make sure to tune in. You definitely do not want to miss that. And once again, thank you so very much to our presenter, Sharon, and to all of you for tuning in. And so from all of us here at the Invasive Species Center, have a wonderful day. Take care, everyone.